Ma'am, we wrote translation. It takes place in the ribosome. We have to write from ribosomes of nucleus.
Oh. Okay, so did I discuss the steps of translation with you all? No, right? Okay. So today we're going to talk about the steps of translation. Now, see, we know that the process of translation, it takes place after the transcription has been completed. And apart from that, in the steps of translation, the first thing is that activation of amino acid takes place. Now, why and how does this happen? Because to drive out the whole entire process, the amino acids are extremely important. Why? Because the proteins are formed when the amino acids, they come together and get joined by the peptide bond to make a polypeptide. So for that very reason, it takes place like the activation of amino acid. It takes place when an amino acid binds with the ATP, the first step. Now, after that, in the second step, what happens is the charging of tRNA or amino acylation of tRNA, meaning when an activated amino acid gets linked to the tRNA. Now, the amino acid that has been activated, it will get linked to the tRNA. And since amino acid is getting linked to the tRNA, that is what we call as the charging of the tRNA. Now,
Ma'am, can I
after
off my camera? I can't hear you. Can I off my
Ma'am,
camera? I can't hear you clearly. My
okay,
internet is not
okay.
working.
ma'am,
Yes, Khadija.
I'm actually outside right now, so I can't on my video.
Okay, it's okay. It's okay. All right. So yeah. So as I was talking about, first step of translation is when the amino acids, they get charged, or you can say they get activated. And when do they get activated? They get activated when they bind to the ATP. Now after that, the charging of tRNA takes place, or this whole process wherein the tRNA gets charged, we can call it as the amino acylation of tRNA. So when an activated amino acid gets linked to the tRNA, it is called as charging of tRNA. And lastly, for the third step, formation of polypeptide chain takes place, meaning this ribosome, they act like a catalyst. Now, the 23S rRNA in bacteria is the enzyme called as ribozyme. Now, a translational unit in the mRNA is the sequence of RNA that is flanked. Flanked means like at the start position, there is the start codon, and at the end position, there is the stop codon. So flanking means in the beginning, in the end, there are things to be put. Now, the translational unit, it codes for a polypeptide, and we know that a polypeptide is a protein. Now, an mRNA also has some additional sequences that are not translated, and those sequences of mRNA that do not get translated, we call them as the untranslated region, UTRs. Now, the UTRs, they are present at both 5' end and they are present at the 3' end. Now, when the untranslated region, they are present at the 5' end, so they are present before the start codon. And when it is present at the 3' end, so it is present after the stop codon. So this is the location of those areas in the particular mRNA sequence which will not get, you can say, translated, or which will not get made into protein. So those are the UTRs, that is untranslated regions. Now, initially, these are, you can say, the before steps that take place. Now, moving forward, what happens is that after the flanking and the charging of amino acid and everything is done, now the translation actually takes place like chain initiation, chain elongation, and chain termination. 3 stages. Now, for the process of initiation, the ribosome, it comes and binds to the mRNA, because we know that when the transcription has been completed, the mRNA gets released, right? Now, this mRNA is what is going to turn, or you can say produce the protein, the end product. So, for the process of initiation, the ribosome, it binds to the mRNA at the start codon, meaning at the AUG. Now, this is recognized only by the initiator tRNA. Now, ribosome starts the elongation phase of the translation. And in this phase, the complex of the amino acid along with the tRNA, meaning this charged tRNA, it starts binding to the codon, which is present on the mRNA, and then starts forming the base pair. So that throughout the length of the mRNA, the base pair formation takes place. Now, the ribosome, it moves from codon to codon all along the mRNA, meaning it moves from one codon to another codon and so on throughout the entire length of the mRNA. Now, in this way, the amino acids, they are added one by one, and as they are getting added, they are getting translated into the protein. Now, at the end, meaning when the whole entire mRNA length has been translated, then a release factor comes along and it binds to the stop codon. And then the translation is terminated or it is ended. Now, what we have is that a complete polypeptide. And remember, whenever it is written polypeptide, we are meaning the protein which has been made from the mRNA. So, after it has been made, then it is released from the ribosome. This is the whole entire process of translation. First, these are some additional steps that take place. First, activation of amino acid. Then the amino acid binds or gets linked to the tRNA, that is charging of tRNA. And then the formation of polypeptide chain takes place. Okay, and then it gradually, you can say completion of the whole entire protein synthesis, it takes place after chain initiation, chain elongation, and chain termination. Lastly, when the whole entire three processes are done, then a release factor comes along, and with that, the process gets terminated. Finally, the polypeptide, which was being formed inside, then now it gets released from the ribosome. Is it clear? Any doubts?
No.
No, ma'am.
To you all? So with this, we have actually
Yes, ma'am.
completed the translation process. Now see students, on this Tuesday, I think you people have your test. So I'll tell you a portion which I have given. Now see, as the chapters have been completed one after one. So I was thinking let's do a quick revision also in the test. So in this test, the upcoming test, you people have sexual reproduction in flowering plants, human reproduction, and then molecular basis. The first two chapters, we are going to revise that, and this is a new chapter. So three chapters are going to come in your test. So you people can note down that sexual reproduction in flowering plants, human reproduction, and molecular basis of inheritance. Three chapters are coming for your test. So prepare well.
Ma'am, in molecular basis of inheritance, till where do you have to study?
Till the thing that I'll be completing today. For example, let's just say till translation. Okay? Till
Okay,
translation.
ma'am.
Clear to you all? I'll not ask anything after translation, but you have to study whatever it is, like however many lines are there till translation. Okay? Ikra, were you saying something?
No, ma'am.
Okay. So that is your portion. Now, do you people have any doubts up until now?
No, ma'am.
No, ma'am.
All right. Now, after this topic has been completed, there is one topic that requires some understanding, and that is lac operon. Then there is human genome project. This is all theory based. So I think I'm going to use NCRT PDF so that you people can understand it better. Okay, first you people note this down, then we'll move on to the NCRT PDF. What was the last thing that you people have written?
Ma'am, translation.
Translation
It
takes
takes
place in ribosome.
in the ribosome. Yes, ma'am. It takes place in the ribosome.
All right. Yeah, you can note it down. You can switch off your video and write down. Done.
Done, ma'am.
All of you are done?
Yes, ma'am.
I'm done. Everyone's done?
1 minute, ma'am.
Okay.
Done, ma'am. Done, ma'am.
Everyone's done?
Yes, ma'am. Done, ma'am.
Done, ma'am.
Ma'am, done.
Ma'am, done, ma'am. Done, ma'am.
Everyone's done?
Yes, ma'am.
All right. Okay. Just give me 1 second. Can you see the screen, students?
Yes, ma'am.
Yes, ma'am.
Okay. All right. So we had discussed up until translation. And now the things that are there, they're mostly theoretical. They're mostly like you don't need extra knowledge apart from what NCRT says. So we're going to pay attention to this. Okay, so
Ma'am,
now,
should I also get my textbook? Are we gonna underline this or are we gonna write down this in our
yeah, you
notebook?
people can get your textbook. All of you have your textbooks by your side. So now the next topic that we had to discuss is the regulation of gene expression. Now see, gene expression, it means that whenever a particular, you can say, synthesis of protein is done, that is what we refer to as the gene expression. Now, or the gene expression and its regulation, what happens is that it happens in the case of eukaryotes at so many levels, such as the transcriptional level. We know that during the transcription, what happens? mRNA is formed, right? Now, at that point of time, we can call it as regulation of gene expression also. Then processing level, we know that after the RNA has been formed, it is initially the hnRNA. Then that hnRNA gets transformed into the mRNA. And we know that there is splicing, there is capping, tailing, all of these things are done. Then the mRNA, which has now finally been processed from the nucleus, it travels into the cytoplasm. Why? Because the translation takes place in the cytoplasm. And then at the last, the translational level is finally when the proteins are synthesized. That is also when gene regulation happens. Now, the genes in a cell, they are expressed to perform a particular function or a set of functions. Each gene that codes for a protein, let's just say an enzyme or anything, they have their own functions to perform. For example, if we talk about the enzyme that is beta galactosidase. Now, this enzyme is synthesized by E. coli. Now, what it does, this galactosidase, it breaks down the lactose, which is a disaccharide. If lactose is a disaccharide, that means that it is made up of two sugars. So every lactose molecule is made up of galactose and glucose. Now the bacteria use them as a source of energy. Now if the bacteria, they do not have the lactose around them, then they would no longer require the beta galactosidase. Obviously, now if a person is not eating a lot of protein, then the protein digesting enzymes will also be not required in that person. Same goes for E. coli. When the E. coli does not use lactose, then why does it require the beta galactosidase? Of course, it won't require. Therefore, in simple terms, it is the metabolic, physiological, or environmental conditions that regulate the expression of genes. Now, again, the genes are expressed as, or you can say, according to what the need of the any particular organism, be it bacteria, be it us eukaryotic organisms. So it is metabolical, meaning whatever reactions are taking place inside our body, whether it is anabolic or catabolic. Then physiological, according to the body physiology, what is the requirement? Then that again goes for the regulation of gene expression. And then also the environmental conditions that play a big role in that. Now the development and differentiation of embryo into adult organism are also a result of coordinated regulation of expression of several sets of genes. We know that once like the fertilization has been completed, the zygote has been formed. Now that zygote slowly starts developing into an embryo, and then that embryo develops into a fetus, and later so on, all of these stages of life take place. But again, do you think will it be possible without the genes and how the genes are regulated? Do you think will it be possible? Yes or no?
No, ma'am.
Of course not. Without genes, or we can say anything that happens in our body, or let's just say if we require any enzyme at a particular point of time, if we require any antibody, all of these things, they happen because of the gene regulation only, because the whole entire process of protein synthesis goes through that. Now, in prokaryotic organism, the control of the rate of transcriptional initiation is the predominant site for control of gene expression. For example, if the gene expression, if we have to stop it, then the transcriptional initiation, meaning the point where initiation in the transcription takes place, if we stop things then and there, then no more gene expression will carry on. So that is the site where we can actually stop or start. Now, in a transcription unit, the activity of RNA polymerase at a given promoter is in turn regulated by interaction with accessory proteins. You people remember activators and repressors? Right? Sigma factor and rho factor, you remember? So why?
Yes,
Those are
ma'am.
accessory proteins. Those are additional stuff that was coming on to play in the transcriptional process. It was not like the basic necessity, but it is something extra that plays a very important role in the process of transcription. Now, these regulatory proteins can act both positively, meaning it can activate the whole entire process, or negatively, meaning as repressors, meaning to stop. Now, the accessibility of promoter regions of prokaryotic DNA is in many cases regulated by interaction of protein with sequences termed operators. So now we're going to step into the world of operator and understand what does it mean. Now, operator region is adjacent to the promoter element in most operons, and in most cases, the sequence of the operator bind a repressor protein. Now, particularly if we want to study the expression, or you can say regulation of gene expression in the case of prokaryotes. So we'll talk about lac operon. Now, lac operon, lac stands for lactose. Now, for example, lac operon is present only in the lac operator is present only in the lac operon, and it interacts specifically with lac repressor only, because it is like, as I told you, that every enzyme, every amino acid is very specific with its function. Same goes for the operator. Now talking about the lac operon. Now, the elucidation of the lac operon was also the result of close association between a geneticist, that is Francis Jacob, and a biochemist, that is Jacques Monod. So Jacob and Monod, they gave the lac operon. So remember that, Jacob and Monod. Now, they were the first to elucidate a transcriptionally regulated system. Now, in lac operon, here lac refers to lactose. A polycistronic structural gene is regulated by a common promoter and regulatory gene. Such arrangement is very common in bacteria and is referred to as an operon. So, operon is a proper regulatory system that is present in the case of bacteria. And we are going to talk about how the gene regulation, or you can say expression, takes place in bacteria, which is a prokaryote. Now, just like how lactose operon, lac operon is there, same way TRP operon, that is tryptophan operon, HIS operon, that is histidine operon, val operon, valine operon. Same way, all of these different kinds of operons are present, and they have their different, you can say, inducers or regulators. Here we are going to study about the lac operon, which is the most important one out of all of them. Now, the lac operon, it consists of one regulatory gene, that is the I gene. Here, the term I does not refer to inducer, rather it is derived from the word inhibitor. So, what do we have? We have the regulatory gene. Now, apart from the regulatory gene, what do we have? We have the structural genes that are 3 in number, Z, Y, and A. Now, let us see what are, like, every gene will code for something. So, let us see. The I gene, it codes for the repressor of the lac operon, meaning if the I gene synthesizes its material or synthesizes what it's supposed to, then the whole entire lac operon process will get repressed. It will get stopped. Now, the Z genes, they code for beta galactosidase. Okay, each of these genes have their own functions to perform. The I gene is responsible for coding for the repressor. Then the Z gene. The Z gene codes for the beta galactosidase, which is primarily responsible for hydrolysis of the disaccharide lactose into its monomeric unit. So remember, we talked about that beta galactosidase. It breaks down the lactose into galactose and glucose. So this is that beta galactosidase, and it is coded by the Z gene. Later on, the Y gene, it will code for permease. What does it do? It permeability. It has things to do with the permeability. So it increases the permeability of the cell to beta galactosides. Now, the A gene, it encodes for the transacetylase, meaning each and every gene has been responsible for a given function that it has to perform, meaning a set of genes that it has to make. Hence, all the three gene products in the lac operon, they are required for metabolism of lactose, because we are talking about how lactose operation takes place. Now, in most other operons as well, the genes present in the operon are needed together to function in the same or related metabolic pathway. So, I gene codes for the repressor, Z codes for beta galactosidase, Y codes for permease, and A codes for transacetylase. This much is clear to you all, students?
Yes, ma'am.
Yes, ma'am.
Any doubts? I'll give you the notes. Don't worry about that. First you understand, I'll give you the notes, okay? Any doubts up until now?
No,
No,
ma'am.
ma'am.
All right, very good. Now see, this is the whole entire diagram, and many a times this diagram is asked in the question also. Now this I, it codes for repressor, right? This is the I that we were talking about. It will code for a repressor. Then there is the O, which is the operator region, and then Z will code for beta galactosidase, Y will code for permease, and A will code for transacetylase. Now, when I will code for the repressor, okay, then what will happen? This repressor will come and bind to the operator area, and it will not let transcription happening. It will not let the process to continue. So whenever the repressor has been synthesized by the I, then what will happen? The repressor binds to the operator region and prevents RNA polymerase from transcribing the operon, meaning transcription will now not take place, meaning mRNA will not be synthesized. But whenever inducer is present, then what will happen? This I, which is the repressor gene, it will make repressor mRNA, and this is the repressor. Now, in the presence of an inducer, the inducer comes and binds to the repressor and makes it inactive. So this repressor does not go and bind to the operator. So now the transcription process continues, and then translation also happens. So this is what the lac operon in bacteria works like. Then the lac mRNA is synthesized. Always remember that inducer, whenever inducer will be present, it will go and bind with the repressor molecule and make it inactive. Now, once the repressor molecule has been made inactive, then it will not bind to the operator. Clear to you all?
Yes,
Yes,
ma'am.
ma'am.
Any doubts?
No, ma'am.
No, ma'am.
All right. Now, lactose is the substrate for the enzyme that is beta galactosidase, and it regulates switching on and off of the operon. Hence, it is termed as the inducer, meaning whenever lactose is provided to the bacteria, then it will be transported in and it will be sent by the action of permease, right? And then it will induce the following, meaning this inducer that we are talking about, this is the lactose molecule. Clear, students? Whenever the preferred source, that is glucose, will not be there, then lactose will be working. Now, the repressor of the operon is synthesized all the time constitutively from the I gene. The repressor protein binds to the operator region and prevents RNA polymerase from doing the transcription process. In the presence of inducers such as lactose or its other forms such as allolactose, then what happens? The repressor is inactivated by the interaction, because the repressor, like the inducer, comes and binds with the repressor. So the whole process of transcription then happens normally. So essentially, regulation of lac operon can also be visualized as regulation of enzyme synthesis by its substrate. Remember, always remember that glucose or galactose cannot be used as inducer for lac operon. Why? Because this is lactose operon. So the inducer will always be lactose. Now, this type of regulation, wherein because of the presence of inducer, the repressor is being inactivated, then only the process continues. So that is what we call as negative regulation. So lac operon is negative regulation. And lac operon is under the control of positive regulation as well, but we do not have to discuss that. But remember that the regulation of lac operon by repressor is negative regulation, because the repressor will come, it will bind to the operator region and transcription will not happen. So just in case if we want the transcription to take place, what do we have to do? We have to make the repressor inactive, then only the transcription will take place. So in this particular diagram is very important. And like while you're writing the answer, let's just say anything about the lac operon comes. If you draw this diagram very nicely labeled and everything along with your answer, you're likely to get very good marks in that particular answer. Any doubts in lactose operon, students?
No, ma'am.
No, ma'am.
All right. Now talking about Human Genome Project. Now see, when people started to talk about genes, when they got more and more knowledge about the genes, then what happened? They started to do bigger projects or bigger works on genes and how the genes are regulated and everything. Now, up until now, we have studied about DNA, which is our genetic material, right? Now, if two individuals, they differ, their DNA sequence should also be different, at least at some places. These assumptions led to the quest of finding out the complete DNA sequence. Now, people wanted to find out how many DNA sequence or what is the sequence of DNA in the humans. So that is what Human Genome Project was. Now, with the establishment of genetic engineering techniques, where it was possible to isolate and clone any piece of DNA, and availability of simple and fast techniques for determining DNA sequences, a very ambitious project of sequencing human genome was launched in the year 1990. You people have to remember the year for this particular project. In 1990, this was launched. Now, Human Genome Project was a mega project. Why? Because imagine one person and one person's genome. It is a very, very huge thing. Now, human genome is said to have approximately 3 into 10 to the power 9 base pairs. Imagine. Meaning how many adenine, thymine, guanine, and cytosine are there in our body. That is what they wanted to find out. Now, if the cost of sequencing required is US $3 per base pair, then the total project will cost 9 billion US dollars. That is why this was a mega project. Further, if the obtained sequences were to be stored in typed form, meaning we have to maintain the data, and if each of the page of the book contained 1,000 letters and each book contained 1,000 pages, then 3,300 such books would be required to store the information of DNA from a single human cell. Only one cell of human being. Normally working on human is not, you can say, allowed. Now, the enormous amount of data expected to be generated also necessitated the use of high-speed computational devices for data storage and retrieval and even analysis. So, Human Genome Project was closely associated with the rapid development of a new area in biology, that is bioinformatics. So, this was the basic, you can say, ideology that went into the Human Genome Project. Now let us see what are the goals, like what were the goals. Some of the important goals of the Human Genome Project were as follows. Identify all the approximately 20,000 to 25,000 genes in human DNA. Then determine the sequence of 3 billion chemical base pairs that make up human DNA. Imagine, this was the huge goal that they had. Then after determining the sequences, like what is the sequence, whether A is there, T is there, G is there, C, like what is the sequence? After that, they had to store this information in the databases. Then also to improve tools for data analysis, and then transfer related technologies to other sectors such as industries. Lastly, to address the ethical, legal, and social issues that may arise from this project. So this was the goal that they had. Now this Human Genome Project, it was a 13-year project which was coordinated by the US Department of Energy and the National Institute of Health. During the early years of Human Genome Project, The Wellcome Trust UK became a major partner, meaning a lot of investment was also required. Contributions from Japan, France, Germany, China, and other countries were also involved. This project was completed in the year 2003. Knowledge about the effects of DNA variations among individuals can lead to revolutionary new ways to diagnose, treat, and someday prevent thousands of disorders that affect human beings. Like in many of the human beings, because we have studied that principle of inheritance chapter, in that we have seen there are so many genetic disorders that can take place in a human being, right? So if a person knows why the disorder is taking place, in which particular gene the disorder is taking place, then what will happen? Maybe the person can be protected from those disorders because we know the sequence. Now, besides providing clues to understanding human biology, besides providing understanding human biology, learning about non-human organisms, DNA sequences can lead to an understanding of their natural capabilities that can be applied towards solving challenges in healthcare, agriculture, energy production, environmental remediation, all of the above. Many non-human models, yes, these are some of the things that can be asked in the question, like what were the non-human models that were also considered? So bacteria, yeast, Cenorhabditis elegans, which was a non-pathogenic nematode, and Drosophila, which is the fruit fly, the plant, which is rice, and Arabidopsis. All of these have also been sequenced apart from the human. Okay, so any doubts up until now, students?
No,
No,
ma'am.
ma'am.
Okay, just give me 1 second, just 1 second. All right. Now coming on to the methodologies, how this was done. So the method involved two major approaches. One approach focused on identifying all the genes that are expressed as RNA, meaning every single gene that is present in our human body, such as in the form of RNA. And we refer to as the express sequence tags, ESTs, meaning all of the genes that are expressed as RNA. The other took the blind approach of simply sequencing the whole set of genome that contain all the coding and non-coding sequence, and later assigning different regions in the sequence with functions. This is known as sequence annotation. So the methodology for Human Genome Project were two: express sequence tags and sequence annotation. Now, what happened for the sequencing process? The total DNA from a cell is isolated and converted into random fragments of relatively smaller sizes. Okay, why? Because DNA is a very, very long polymer. It is like 2 meter long per. So the technical limitations are there. So that is why they have to be done into certain fragments. And then they are cloned in suitable host using specialized vectors. Cloning is done, and cloning means you can make multiple copies of one thing, so that is cloning. So we do the cloning of each piece of DNA fragment, and then the sequencing of it is like done by express sequence tag and sequence annotation. The commonly used hosts were bacteria and yeast, And the vectors were the bacterial artificial chromosome and yeast artificial chromosome. See, here you are seeing many new words such as cloning, then vectors, all of these things. You will learn about this, but in biotechnology, when you will come to that chapter. For now, for as far as this chapter goes, just learn, okay, these were the vectors. Just learn that, okay, these were the hosts. Clear,
Ma'am,
students? Hmm?
for that chapter biotechnology, is this chapter also some terms from this chapter will also be used there?
No. But some of the like terms that you are seeing here, like cloning, then PCR, all of these things will be used there. Meaning they will be taught there. Here they have just used it. You will be taught in that particular chapter. Okay. So it's very interesting. It's very awesome chapter. You will like enjoy it a lot. So don't like stress on it. You can just learn that, okay, these are the names of the vectors. These are the name of like the host and everything. Okay. Now, the fragments, they were sequenced using automated DNA sequences that worked on the principle of a method developed by Frederick Sanger. Now, he was the person who developed the sequencing method. Now, these sequences were then arranged in some overlapping regions present in them. This required generation of overlapping fragments for sequencing. Alignment of these sequences was humanly not possible. Therefore, specialized computer-based programs were developed. Now, these sequences were subsequently annotated and were assigned to each chromosome. The sequence of chromosome 1 was completed only in May 2006. This was the last of 24 human chromosomes, 22 autosome and X and Y to be sequenced. Now, another challenging task was assigning the genetic and physical maps on the genome. Now, this was generated using information on polymorphism of restriction endonuclease, recognition sites, and some repetitive DNA sequences known as microsatellites. Again, these things will be, you can say you'll understand. Now, this one about microsatellites can be like answered in DNA fingerprinting. But up until now, have you people understood everything? That this basically the whole
Yes,
entire
ma'am.
sequencing process was
Yes,
being done by a computer. Okay. Now let us see what are the features. Like after the human genome project was completed, let us see what are the you can say features of human genome project. Now the first feature. Human genome contains 3,164.7 million base pairs. These many base pairs are there in human genome. Now, the average gene consists of 3,000 bases, but sizes vary greatly, with the largest human gene being dystrophin. Dystrophin is the largest human gene, and it has 2.4 million bases. Now, the total number of gene is estimated at 30,000, which is much lower, like uh, than the previous estimate. Like people had made sort of many estimations, but it finally came down to this particular number. Almost 99.9% nucleotide bases are exactly the same in all the people, and we only differ from that 0.1%. The functions are known for over 50% of the discovered genes. So genes that are like 50% of them, we know the functions, what they are supposed to. Out of them, less than 2% of the genome, it codes for proteins. Repeated sequences make up very large portion of human genome. Like a lot of time, like many repetitions are there throughout the human genome that is present. Now, the repetitive sequences are stretches of DNA sequences that are repeated many times, sometimes 100 to 1,000 times. They are thought to have no direct coding function, but they shed light on chromosome structure, dynamics, and evolution. So they are not directly involved in coding, like not all the repetitive structures are performing the function in coding, but some of them are. Chromosome 1 has most genes, that is 2,968 genes, and the chromosome Y, the sex chromosome, it has the least gene, that is 231 genes are present. Now, scientists have identified about 1.4 million locations where single base DNA difference, that is SNPs, SNPs, that is single nucleotide polymorphism, occurs in human, meaning difference by 1 base pair. This information promises to revolutionize the process of finding chromosomal locations and disease associated sequences and tracing human history. So basically, up until the point number 8, these are the findings or the features that the Human Genome Project offered. Any doubts, students? These are just data. You have to remember the data. Just remember like this point, this point. Then how many number of genes are there, the estimation finally, and then this point. These are very important. Okay. Now coming on to the application and future challenges. Now deriving meaningful knowledge from DNA sequences will define research through the coming decades, leading to our understanding of the biological systems. As I told you that if we understand the human genome, how it works, what are the sequences, then it can be very beneficial in the health science particularly. Now, this enormous task will require expertise and creativity of tens of thousands of scientists from varied disciplines in both public and private sectors. One of the greatest impact of having the human genome sequence may well be enabling a radically new approach to biological research. In the past, researchers studied one or a few genes at a time. With whole genome sequences and new high through output technologies, we can approach questions systematically and on a much larger or a broader scale. They can study all the genes in a genome, for example, all the transcript in a particular tissue or organ or tumor, or how tens of thousands of genes and proteins work together. All of this, basically how the chemistry inside our body is there, that can also be studied because of the knowing or the knowledge or the data that is gathered from the Human Genome Project. Okay, students, any doubts?
No, ma'am.
No, ma'am.
Clear? All right. So
Yes,
now
ma'am.
only one topic is left, that is DNA fingerprinting. I'm thinking to do it in the next class. Are you people like your brain is fogged up now? Or you people have the capacity?
Very soft, ma'am.
Brain is fogged up? All of yours?
Yes, ma'am.
Okay, yeah, I was thinking the same. So like only one topic is left, and then I have to give you the notes for Lacopron. That is very important. Okay, so I'll give you the notes for Lacopron tomorrow, and rest all these Human Genome Project and nothing, these notes are not required. You just are to study whatever is there in the NCERT. Just read it and learn it. That is all there is to know. But Lacopron, I'll give you notes about that. Okay. So let's just keep it till here only for today's class. And in the next class, we'll discuss DNA fingerprinting, meaning tomorrow. And then